Hi everyone and welcome to GCSE Revision with MFS. In this video we look at the required practical for food tests. We focus on the method used to carry out this required practical and the key observations you would see. Please ensure you hit the subscribe button and the notification bell to ensure you never miss a video from GCSE Revision with MFS. If you would like to join our live lessons, the link to book your place is in the description below. Okay, we will make a start now. So welcome everyone to our GCSE Biology lesson. In today's free lesson, we are going to be looking at the required practical for food tests. So this is part of Unit 2, Cell Organization, and it's one of the key required practicals that you need to know. Again, this is a common exam question. It normally tends to pop up commonly on exams. So it's a required practical as well. Make sure you learn the details. So on the slides, I've got the method and I've got the results. So food test is one of the harder topics that are in the GCSE biology spec because there is a lot of key detail you need to know. Okay, so in today's lesson, the purpose of today's lesson is to go through this required practical. Okay, so if we make a start. Okay, so food test. Now, before we move on to the method, food test is just a way of determining in a food sample whether there is protein present, whether there is starch present, whether there is lipids present. Okay, so it's a way to determine what is present in a food sample. So it's the way to determine what is present in a food sample. We will go into more detail in this lesson, but if I just show you an overview, we have a beaker, which is just like a cup. You place your food sample in the beaker, and then you add sort of a solution into it. Okay, so you add a solution into the food sample, and then it will turn a specific color, and that will tell you if what you're testing for is present in it. For example, if I give you a really rough example here, let's say we are testing for the presence of starch. Okay, so starch we've got there. Let's say we are eating some food and we take a sample of that food. Let's say we've got bread. Okay, so this is our bread. And we want to test if this bread has starch present in it. Okay, so first of all, what we would do is we would break the bread down into small pieces. Okay, so we would just break the bread down into small pieces, such as this. Okay, so this is our bread in small pieces. We've just broken the bread down. And then we then place it into a beaker. Okay, so we've got a beaker. We just place the bread that we've broken down into the beaker. So now the beaker contains the small pieces of bread, as you can see. Then we add a solution into it. So to test for starch, you would use iodine solution. So iodine solution is used to test for starch. We will go through this in this lesson as well. I'm just giving you a brief overview of how to carry out a food test. Iodine solution is used for to test for starch. Okay, because we are testing for starch, we would add in iodine solution into the beaker, a few pipettes of iodine solution into the beaker. Now the beaker, you would see, would look something like this. We've got our bread in small pieces in the beaker already, and now we've added sort of iodine solution into it. Okay. Now this solution will now change a specific color. Okay. If starch is present, it will change color. Okay, so it will change color if starch is present. And that's how you know starch is present, because it will change color. However, if it doesn't change color, that means starch is not present. So if there is no change in color, no starch is present. Okay. So I've just given you a brief overview. So normally what you have is your food sample. In this case, we're using bread as the example. You break it down into small pieces and you place it into a beaker. Then you add in a solution and then it will change. Sorry about that. It will change to a specific color to show a positive result. And if there is no change in color, then starch is not present. So a positive result, starch is present, means that the solution will change color. However, if no starch is present and a negative result is there, you won't see a change in color. So now we'll move on to the method. So in terms of scientific equipment, in order to grind the food products, let's say we've got this bread, instead of breaking it down, what we would do is you would grind it. So you would grind it using a pestle and a mortar. So a pestle and a mortar is something like this. So you just have this sort of a dish over here, and then a mortar is something like this. You place your food sample into it and then you grind it with this mortar. Okay. And that's how you break down the food pieces into smaller food molecules. So let's say we're testing for bread. In this case, we would place the bread into this dish and then we would grind it using that equipment until it's into small pieces. Okay. And then you transfer the products into a beaker and add some water as well as stirring the solution. So once this is ground up, okay, so once this is ground up into small small molecules, such as this one, this is small molecules, we then place it into a beaker. So then we then take it and place it into a beaker, the small food molecules. Okay, we just place it into a beaker. Okay, and then you just add in some water as well and start stirring the solution. So you want to see a color change. That's why you need to make this solid into a solution. So you just add in some water, okay, and that makes it into a solution so you can see the color change more visibly. So into this, you have your food molecules that you've just broken down using a pestle and a mortar, and then you would just add in water. So add water, and the reason you add water is to make it soluble, and so you can see a more visible color change. Okay, so because iodine solution is basically liquid, when we're adding it into another solution, you can see the color change more visibly. And that's why we're just adding water into it, so the food molecules dissolve in, and when you're adding the indicator you want to test for, you can see a more visible color change. And now filter this into a conical flask, and then the results are put on the next slide. So basically what you want is that any insoluble food molecules to be left out. Okay, so once we've added the water in, we want all of the food molecules, we want them to be as small to dissolve into the water. So we can filter out any other big food molecules, so we're just left with the solution. So once we've added water, what we're doing is that we are filtering. We are filtering, so the large pieces of bread, in this case, the large pieces of the food molecules, they will be left out and we will just be left with a solution of our food sample. So after filtering, we would use a filter paper, filter funnel, pour this solution into it and place a conical flask underneath it. And you would see that the insoluble food molecules will be left behind and the soluble stuff will fall through. And then you will be just left in your conical flask with a solution. And this solution will contain your food sample. Okay, so I'll just draw a conical flask there. Okay, so you filter out this over here, so you're just left with the soluble solution. So you will just be left with the solution in your conical flask. Okay, because we don't want any big food molecules to be interfering with our experiment. Okay, so if I summarize off this slide, what we've actually done is we have using a pestle and mortar. I'll highlight the key points as well. Using a pestle and mortar, we have ground up the food products so they're really small, such as this over here. We're using bread as the example. Okay, and then this is the sort of equipment, pestle and mortar, you would use to grind it up, and then you transfer your small food molecules into a beaker over here, just like that. Okay, and then you add in some water so it dissolves in. You have to stir it using a stirring rod, most preferably using a stirring rod, and you would see that the small food molecules will dissolve into the solution. And then you would filter out the solution to remove any big food molecules, because we don't want big food molecules in our experiment. You filter out the big food molecules, so you're left with just a solution in a conical flask. As you can see there, just a solution. And now you are going to add in the indicator. In this case, we're adding iodine solution to this solution, and you will see a color change if starch is present. Okay, so you transfer the products into a beaker, add some water, stir the solution. Then you filter this into a conical flask, and then we're going to test for the results. Okay, and here are the results. So that you can test in this required practical, you will test for starch, sugar, protein, and lipids. These are the 4 groups you will test for. And this is why this is a difficult required practical, is because you basically need to learn this table over here. You will need to learn the results and the test. So everything that's present in this table, you will need to know for the GCSE exam. So as we were discussing over here on this slide, we were discussing the matter of bread and iodine solution. Okay, so if I just draw this out once again, so we've got our sort of small food molecules, in this case bread. We've just used a pestle and mortar to grind up our bread. Now our bread is in small pieces. We then add in some water so it dissolves in, and then we filter it out into a conical flask. So we're just left with a solution with small small molecules of our sample. Okay. Now, once we are up to this point, what we would do is, because we're testing for starch, we would add in iodine solution. Okay, so we would add in iodine solution to this conical flask. And if you see a color change, if the solution changes to blue or black, you know that starch is present. So if solution changes color to blue or black, then you know that starch is present. However, if there is no color change, then starch is not present. Okay? No color change, then you know that starch is not present. And that's how it is for all of the tests. If we were testing the sample with sugar, for example, what we would do is that we would add in Benedict's solution this time, because we're testing for sugar. We would add in Benedict's solution to this sample. So we would add in Benedict's solution. However, the only difference this time is that you would need to heat it into a water bath. Okay, so a water bath is just a way of heating the solution so you can see it. So you would just place this conical flask into a water bath after adding Benedict's solution, and then you will see a color change. And the color change for a positive result, so all of these are positive results. So if sugar is present, you will see a color change to green, yellow, or brick red. Okay, so just to reiterate that, we have our food sample in our conical flask. Okay, we have our food sample in our conical flask. If we wanted to test for sugar, we would add in Benedict's solution. And you would need to heat it. Okay, and you would need to heat it in a water bath, just so you have it's warm enough for a reaction to happen. And for a positive result, so if sugar is present, you would see a color change to green, yellow, or red. And this is for a positive result. If sugar is not present, there is no change in color. Okay, if we look at proteins, let's say we've got our food sample over here. We've carried out all the previous steps. We've got our food sample, and we want to test for the presence of protein. This time you would have to add in Biuret solution. So you would add in Biuret solution, and you would see a color change to purple. So if protein is present, you would see a color change to purple. And if protein is not present, you will see no color change. If we were to test for lipids, again, you have your solution in your conical flask. You would just need to add in ethanol. And after you've added ethanol, you should see a white cloud emulsion forming. So a positive result will show you that a white cloud emulsion is forming. So a cloudy layer at the top sort of forms, and that is for a positive result. Okay. However, if there are no lipids present, there will be no change in solution. So my advice for you is just to learn this table. This table here is essential. Okay, so testing for starch, you would need to use iodine solution, and a positive result will change the color to blue or black. If you're testing for sugar, you would need to use Benedict's solution, and you would need to heat the solution in a water bath, and a positive result changes color from green, yellow, or brick red. And protein, you would need to use Biuret solution, and a positive result would change color to purple. And to test for lipids, you would need to add in ethanol, and a positive result would show a white cloud emulsion forming, which is just a cloudy white layer forming at the top of the solution. And if there is no change occurring, then that means that that food group is not present. Okay, so if there is no change, then food group is not present. Okay, so basically I've gone over there everything you need to know for this required practical. Okay, so again, if I'm just going to summarize this required practical now, and I'll give you guys a chance to ask any questions in the Q&A. So the food test required practical is just a way to determine what is present in a food sample. And the tests that you have are these 4 tests: starch, sugar, protein, and lipids. You can test for these 4 food groups and see if they're present in an unknown food sample. So first of all, whatever food sample you have, you need to grind it up first so they're small enough so you can test for them. And to grind them up, you would use a pestle and a mortar. And then you would transfer the products into a beaker, add in some water and stir it so it dissolves into the water. And you don't want any large products in the solution, so you need to filter out the large products. And so you're just left with a solution with small soluble products of your food sample. So we looked at the example of bread. We will need to grind up the bread using a pestle and mortar. So it's small enough, transfer it to a beaker, add in some water, stir it so all of it's mixed in with the water. And then you would need to filter it out so any insoluble molecules, any large molecules stay out. So we're just left with a plain solution of our food sample. Then you, depending on what you're testing for, let's say we wanted to test for starch, you would need to add an iodine solution into the solution. And if it changes color to blue or black, then you know that starch is present. If you wanted to test for sugar, you would add a Benedict solution to our food sample and heat it in a water bath. And if the solution changes color from green to green, yellow, or brick red, then you know that sugar is present. And just if I tell you there are 3 colors here in this case. It depends on how strong the sugar is. It depends on how much sugar is present. The more sugar is present, so the redder the color, the more sugar is present. Okay. So the redder the color, the more sugar is present. To test for protein, you would just add in Biuret solution and you would see a positive result if it changes purple. And finally, for lipids, you would just add in ethanol, and for a positive result, you would realize that a white cloud emulsion forms. Okay, so I've just gone through that practical there.